Enzo is from Frankfurt, and he's going to talk about network measures. Enzo.
Okay, good. Where is actually the slide? Oh, here, sorry. Great. Okay, thanks very much for inviting me, and thanks very much for being here. I will talk about actually different thing that's in the abstract book, but it's the same and different. It's about brain dynamics, but also about global connectivity. So I will just start with the motivations of this work. At least for me, everybody has different motivations for everything, so I will just tell you what keeps me going to do this. Well, the first observation that I'm going to make is that if you take a drug, a psychedelic drug, you don't need anybody to tell you what you feel. You essentially have a first-hand perspective, a first-person perspective of what's going on. Then you can even theorize about what's going on, and you can build, well, you can make somehow measurements by introspection, and you can gain knowledge, first-person knowledge about the drug without doing any scientific experiment, just by taking it. Of course, it is currently impossible to go from a third-person perspective to the first-person perspective. This is, if you take, for example, Robin's study on fMRI, you have an objective view of what's going on in the brain when you take the drug. But how can you, from that objective knowledge, that those facts, those scientific facts, go to reconstruct what the person is feeling? Obviously, you can't. That's a difficult problem. It's so difficult that actually that's the name of the problem. It's called the hard problem. So, yeah. And however, I say that it was not possible to go from the scientific data to the first-person perspective. But it's possible to go the other way around, to go from the first-person perspective to the scientific data, to the objective data. This is, we have some, there are some features of consciousness that everybody knows, because everybody in this room is conscious, right? For example, you know that you can divide your attention into two things at a time. For example, you need to focus your attention, and well, there are many other characteristics of consciousness that you know about, right? And those characteristics can be changed, reduced during sleep, coma, epilepsy, etc. There are states of altered consciousness. So obviously, you can go from those features of consciousness that you know about to theorize what the objective, the changes are going to be, right? For example, in an fMRI experiment. So, well, my motivation is to go to study different states of consciousness, sleep, coma, etc., to try to relate these subjective features of consciousness with experimental objective data. And the goal of this talk is going to be first that one, to understand the objective neurophysiological changes which follow psilocybin intake. I'm going to talk about essentially the same experiments that Robin did before, but in a different perspective. And then, of course, the idea is to correlate these changes and alterations in the subjective feature of conscious experience with, with objective data. And ideally, at the end of the day, one would like to come up with a relationship between the objective and the subjective features of consciousness. But that's essentially very difficult, especially if we're talking about the causal relationship. So the first part, I'm going to talk about neuroimaging, the psychedelic experience. This is essentially, I'm going to talk very briefly about Robin's result. So this is what Robin just talked about. Here is the decreased cerebral fluid flow, and this is the decreased activations after psilocybin intake. And he said that this came as somewhat as a surprising result. And well, of course, you see here this brain hub, here the cuneus, and also for the gold signal, that you have a decreased activity there. And what does it, essentially this is what this means. You have time here, and you have these measures of activity that you can record with the fMRI scan. And after the infusion, you have that these two things depart, not become different. Here you have the psilocybin, and you have a lower signal, and here you have that the signal remains the same, right? So here I have, in the red dots to show the mean value of one signal. So what essentially is happening is that the signal keeps fluctuating, but around a different mean value. So there are many questions you want to make about this. You can make about this. You found this decreased value of the activity signals, but the signals are still fluctuating around the mean. You see that there are still things going on here. This goes up, down, and also this up, down. So as Robin said before, the brain is never completely shut down. I mean, there are still things going on, and we want to characterize what are the dynamics of these things which are going on, even if the signal decreased. So for 10 years, more or less, all neuroscience, all neuroimaging, essentially was about this mean value here. If the mean value decreases, then we see that there is a deactivation. But it's kind of misleading because it's not a deactivation. Things are still changing over time, so only at a different mean value. So I'm going to first study that. So this is not noise. I mean, if you take this wall signal from during resting state, no task, the subject cells in the scanner. You take the signal from the left and from the right motor cortex, you see that this is apparently noise, but they are coordinated between the left and right hemisphere. So this is certainly not noise. And if you look at all the other parts of the region which have the same pattern, you actually can get the whole motor cortex. These dynamics are very slow, like they fluctuate, they complete one cycle around about 10 seconds every cycle, so they are really slow. And this, you can actually look during resting state at this so-called noise, and only by looking at these spontaneous patterns of fluctuation without any task, looking which parts of the brain have a coordinated fluctuation, you can actually recover many different brain systems: visual, motor, well, auditory, etc. And one of these is very important for today, which is the Informal Network, which is, as well, you already heard about it today, but this is essentially related to introspection, self-consciousness. Also, if people who go into coma or fall into deep sleep, you have decreased activity in this network, so it's certainly related to consciousness. And then it's obviously very important when you are going to talk about the neural correlates of consciousness. So, I'm going to talk about some measures that can characterize these spontaneous fluctuations around the mean. The first one is called variance. Variance is simply the dispersion of the signal around the mean. For example, these are just made-up signals that I created. They are not representing real data. So you have here, right? You have these 5 signals. Here nothing is going on. Here a little bit more is going on, and then it starts to increase. And well, the first one has a variance of 0 because signal doesn't fluctuate; it's constant. And then obviously you have increasing variance. You have from 0 to a higher variance, which the signal is doing a lot of things, right? And one can ask a very simple question: how does the variance of the activity signal changes after psilocybin intake, and where does it change? And when we do that, we find these. This, I'm not going to talk about the statistics, this is significant, so I'm not to bore you with that stuff, but what you find here, this is after psilocybin, a larger variance than before. And this is compared the psilocybin with the placebo. Nothing is found for the placebo, so you will see an empty brain, so I'm not going to put it here. The regions you find with increased variance, you have very clear hippocampus here, and also in the anterior cingulate cortex. The hippocampus is essentially related to consolidated memory. So for example, you have a damage to the hippocampus, you will end up being like this guy in Memento, in which you kind of forget what happened 10 minutes ago. And it's very interesting that you have this increase in psilocybin, and I'll tell you what. First of all, sorry, if you want to look at the patterns, are exactly very precisely located here in the hippocampus, left and right. So, and this is over time. So you have here these 2 left and right hippocampus. You have time here. You have the average blood variance versus time for all subjects with the psilocybin and the placebo. You see that when the injection is done, the left hippocampus and the right hippocampus, you have this increasing variance, and nothing happened for the placebo. And the same for the right hippocampus, and also for the anterior cingulate cortex. So this clearly shows you that the injection of the psilocybin is increasing the variance. So I tried to look at something in literature which gave me a clue of what was going on. I found this paper, in which what they did is they injected LSD to cats, and they recorded neural activity. And what they essentially did was to induce a seizure on the cats. When you have epilepsy, well, that's what they did to the cats. They say that maximal amplitude and longest duration were recorded for dorsal, hippocampal, and entorhinal cortex leads. So they essentially induced seizures in the cats. Actually, I tried to look at the whole article, but I couldn't, because if you look at the date, this is the best I could find. It's 62, like just before they banned LSD, so I couldn't find anything after that. I couldn't, at least. So that thing that what David was talking about, that this really was a huge setback on neuroscience after they banned psychedelic drugs. And this what I show you here, there are two patterns. This one you already saw, and this one here is from another population of subjects, which actually is temporal lobe epilepsy. This was recorded here in London, in Sean Duncan's group in Green Square. And this is exactly the same, the variance. So obviously I'm not saying that taking psilocybin creates seizures, because we know it's not happening, but there are some clues on why these changes in the hippocampus activity could induce changes in consciousness. Because if you think about it for a second, when you have epilepsy, you have a seizure, which is essentially first, in many cases, located to the temporal cortex, to the hippocampus. You have, like, the activity starts to oscillate a lot, an increased variance, right? But if hippocampus is related to the memory, how come that you lose consciousness? Maybe you will become amnesic during a seizure, but during seizures, people do not become amnesic. They fall into the floor, and they essentially lose consciousness. Well, there are hypotheses that there are, due to anatomical connectivity, lots of interactions between the temporal lobe and the fall mode network, which is this network which is very important for consciousness. This was being put forward by, I think, Hal Blumenfeld in Shale, which he says that whenever there is a seizure in the hippocampus, this activity eventually spreads out to the whole network and is changing the quality of consciousness. In the case of epilepsy, of course, you lose consciousness. It's a quite radical change. And there are these changes in the hippocampal and the full mode connectivity. Actually, Robin already talked about them a little bit earlier, and I think there is a poster about that. So there are essentially a relationship between the hippocampus and the full mode network. And then this increased variance is also consistent with a scenario of increased metastability. Metastability, as you heard before, essentially means that the system is more effectively exploring its possible configurations. So if this glass is here, it's not a metastable scenario, but if you, for some reason, you inject energy into the glass and it will start moving around and exploring all the different places in which this glass can be, that's increasing the stability. And this increased signal variance shows that the brain is trying hard to look at possible configurations. Then I look at other measure, which is called the Hartz exponent. It captures the temporal dependency of the signal. That is how different the signals are from noise, because you can have complete noise with a high variance. So here is, actually this is noise in which each of these time points bears no relationship to the previous one. And these signals are becoming increasingly complex, in which that means that at a given time the signal is more dependent on what happened before, and it's not uncorrelated noise. Here is there is an index to measure that, which is the Hartz exponent. It's 0.5 for a completely random signal, and 1 for a signal which is, well, which has certain properties but is essentially very autocorrelated. And this is increased complexity. And then, of course, you can do the same, because if the brain is doing some meaningful processing of, for example, of a sensory signal, you expect that it's not noise what the brain is doing. You expect some structure in activity. And when you do that, you find this increased exponent in the fusiform gyrus. This is also significant. Well, it's for some reason only to the left side of the brain, but it's in the fusiform gyrus, which is very interesting. It's an area which is related to many different features of sensory processing, one of which is very important in this context is color processing. So it's known that the fusiform gyrus is involved in color perception and actually can be activated during hallucinations. For example, during schizophrenia or, well, certain hallucinations which involve color. It's also important in scientists, well, it's also related to some sort of synesthesia, which you see color where they are not, that you are not supposed to see a color. And then I look at the final feature, which is also super interesting, is the low frequency power. So I told you that these important fluctuations during the resting state occur very slowly. One cycle lasts about 10 seconds. And then you can actually look at the contribution of low frequencies, the relative contribution of low frequencies against faster frequencies. So here what I have are also again example signals in which you have a very, very fast, well, essentially this is noise, but you can see it as a fast oscillation, and then you start to increase the importance of the slow oscillations, of slow power oscillations in the signal, and you see that the signal is like that, and then at the end it has a low frequency component here, which essentially makes the mean of the signal oscillate more slowly. So this is increased LFP, which is low frequency power. So I am essentially measuring the contribution of these activity features, which are characteristic of the resting state activity. And when I do that, again, I can look at the brain and see which brain regions show a changed power of low frequencies after and before psilocybin, and I find essentially this, that there is decreased LFP in default mode areas after psilocybin. This means that the signal during, well, after placebo or when you are during the normal resting state, you have a slow oscillation, which is typical of what the brain does in the default mode network during in this area of the network of regions involved in consciousness. And you lose these low fluctuations after the psilocybin intake. This is the contrast before greater than after. So you see that you have this huge blob here in the precuneus, which is a part of this network related to consciousness. You have essentially all the relevant regions in the network involved in this loss of slow power oscillations. This is when you compare before psilocybin and after psilocybin, and you can compare the placebo and the psilocybin, and you get similar set of regions. So what I'm saying is that essentially the activity looks, well, I will tell you in the next slide. I made a nice diagram to show you what's going on, because I think this is fully consistent with what we already know about what the brain does to psilocybin, what the psilocybin does to the brain. So this is from the paper that Robin mentioned in his talk. These are the regions which show a decreased beta power, and this is the power. These are oscillations that you measure directly on the neural activity. So this is not fMRI. You are measuring electrical oscillations that you record with EMG, and you have these fast oscillations in the beta range. These fast oscillations in the beta range decrease their power after psilocybin intake in this set of regions, which looks remarkably close to the default network. And then you also have this other observation here. This is completely unrelated at first. You have the beta band, and you look at which brain regions are related to the fluctuations in the beta band, in these 30 hertz frequencies, and you find essentially the default network, the activity, the volt activity in the default network reflects very closely the beta band you measure with MG. And then at the end, of course, you have the power in the beta band is reflected in slower fMRI oscillations. And then finally, you have this fact that I just told you about, that the low frequencies decrease after psilocybin. So I think we're seeing different sides of the coin here. You can at one side see the frequency in the fMRI, you can at one side see, and the other side see the frequency in the EMG. Essentially the oscillations become slower in general, but you can link those using, well, this observation here. And there is one, regardless of how you look at this, there is one lesson that you learn from this, that you're essentially losing the typical idling resting state dynamic. So there is a characteristic slow oscillation that appears when you are doing nothing in your brain, and this is kind of lost after you take psilocybin, most likely because you are engaged in mind wandering, and exploration of different brain states, and so there is not any more resting state activity, and it also relates to the metastability hypothesis, because again, you are not idling, but you are start exploring different configurations, or possible configurations of the brain. So, this is what I'm going to talk to you about, tell you about the dynamics of the brain, these dynamics of spontaneous fluctuations in the brain after you take the psilocybin. But then my talk has a brief second short part, which is about global connectivity in the psychedelic state. I forgot to write state, well, during psilocybin intake. I wrote here no brain region is an island because you can't really understand the brain just by looking at the brain region. I mean, it's trivial that regions in the brain have to be connected. That's something you don't need a fMRI to learn about. Because you know that, for example, the motor cortex is here, in the middle of the brain, and the visual cortex is on the back, right? And if I see this glass, that's visual information which is processed here. If I decide to take the glass, that's motor information which is processed in another area of the brain. So there has to be an interaction. There's no other way around it. Different parts of the brain need to talk with each other. That's almost trivial thing. So you can't really understand the brain just by looking at regions separately. So actually, this is what I told you before. You have these spontaneous oscillations in the left and right motor cortex. They look like noise, but they are not, because they are exactly the same in the left and the right motor cortex. This means that essentially those regions are doing the same. They are essentially co-activated. They are participating in the same process. So that's called functional connectivity. When you have one region and another region, and you look at what's going on in those regions and they are doing the same, that's called functional connectivity. And that's what Robin told you about in his talk. After psilocybin intake, you have a disintegration of networks. So this is, you observe decreased prefrontal cortex connectivity after psilocybin, and also with the PFC, you have decreased connectivity, so you have two brain regions which show essentially decreased functional connectivity after psilocybin intake. So there are these two networks, the DAS positive network and the Informal Network, are essentially becoming disintegrated after psilocybin intake. So you lose the connectivity in these two networks, which is a very interesting observation. And I thought, well, what happens with all the networks of the brain? I mean, what happens in general? You have the brain, you have lots of different groups of neurons talking to each other, and you want to know, to understand if there is a really global breakdown in connectivity, or this is just affecting these two networks. This is like you lost connectivity at all, well, what's going on? So to do that, I need to understand how, not just the connection of these two regions, but the connections of all regions in the brain, one to each other. So to do that, I will use some conceptualization, which is called network analysis or graph analysis, which I'm very briefly going to explain now, which I call the language of interactions because it's just that. So you defined a graph, which is essentially a group of nodes, which could be people or brain regions, whatever. It's just a point well, just a point, just something. And then you have connections between them, which can be a different type of interactions between these nodes. As an example, for example, all of you which are familiar with Twitter or Facebook will understand this, because Twitter and Facebook are essentially networks. You are one node in the network. Your user is one node in the network. There are other users, which are other nodes. They're connected because they know they are friends with each other in Facebook, or because one follows another in Twitter, but the concept is the same. It's a network of nodes and connections. This is what I'm talking about. This is one node. You have another node and one connection. You have many nodes, many different connections between them. You have another group of nodes which is not connected. So this here and here is like, I don't know, my group of friends and the group of friends of somebody in Malaysia, probably. We won't have any nodes in common. But if I go to Malaysia and get to know somebody, there will start to be, to appear these links between these two groups of nodes. And actually, you have these integrated social groups. And if, of course, if you add more nodes, like we all move to Malaysia, we might end up having just one network. We lose these two different groups of nodes. And there are actually mathematical measures which tell you how segregated this network is in terms of different groups of nodes. There are also other measures. For example, consider this network. This network, you have one node, which will be some of you on Facebook, and you have many different friends, other nodes, right? And you see that you have these connections, and the red connections are the connections among your friends. So you see that these 4 friends have also lots of connections between them. So this network is called to be, is essentially defined as very clustered. So there are measures, which is called a measure which is called the clustering coefficient, which will tell you how many connections occur between different neighbors of a given node. Or more heuristically, if nodes tend to cluster locally in groups, right? So this means that your friends are not only good friends with you, but also between them. Here's another example. You have another network here, you have 2 nodes, right? And you say you want to go from this node to this other node. This could be, for example, metro stations. And you have many ways to go to this node. You can go like this, you could go like this, but there is one way which is the shortest one, which is if you follow the red path. And this is actually something you can measure in networks. This is the average path length, and well, people say that between any 2 persons, there are essentially like 6 acquaintances in between. So if you think about the Pope, well, there might be 6 different persons that if you know him and he knows her, and in the end they know the Pope. Actually, for me, this is probably a lot lower because I am Argentinian and the Pope is also Argentinian. But it's even more funny because probably since you know me, so probably this has, for all the people in the room, this has went down 1 node. So so you are 1 node closer to the Pope, probably. So you can thank me for that, if you want. And well, heuristically, this tells you how accessible are different nodes in the network. So how easily you can go from one node to the other. And actually, this is not just about popes or Facebook or whatever. This can be used for neuroscience because you can actually construct networks using the brain. You have here a parcellation of the brain, which essentially you have the brain and you can identify different pieces of the brain, and then you can look at the signal of the different pieces of the brain, I mean the fluctuations over time of every pair of signals, and you can actually, between every pair of signals, you can compute how similar those signals are. So this is just not in one network, this is between all pairs of regions in the brain. And then you end up having this, which is called the correlation matrix, which I'm not going to explain, but it's simply these rows, it has rows and columns, and every row and column is a brain region, and in the intersection of 2 of them, it tells you how strongly these 2 regions are interacting. If this is high, it means that those regions are doing very much the same. If it's low, it means that, well, one doesn't care much about the other. And then there is some, you can actually go from here to here by saying, okay, all regions which have an interaction which is higher than, I don't know, 0.5 to say something, well, we will consider them as connected. So after you do this, you end up with this, which is essentially an abstraction of the brain in which you have lots of connections, and they tell you, well, the brain's network, how it is constructed. Remember that you have this step here which introduces a technical free parameter, but we won't worry about that. No. So obviously I'm telling you about this because I wanted to apply this to the psilocybin data to understand the global connectivity properties of the brain. And if we start with the average clustering coefficient, which tells you if the nodes are very clustered, you have here, well, this is the free parameter I was telling you about. It doesn't matter, it's just something you can change. But the important thing is that if there is a difference, you want the difference to be over an extended range of these parameters. And then I plot here the placebo after and the placebo before, and you see there are no differences. But then, after psilocybin, you have an increase over the whole range, and also when you compare psilocybin directly versus placebo. And this means that the networks are more clustered, so the regions tend to work more locally connected to each other. Then you have the average path length, which tells you, well, how easily can I go in this network from one node to another. And again, nothing happens for the placebo, but interestingly, this parameter goes to a higher value after psilocybin. So this means that it's no more, you you have more nodes in between any two pairs of nodes. So it's more difficult to go from one node to the other. It's a lengthier path, right? You can understand it like that. So this is very much in line with what Robin found, because this means some sort of disintegration. I mean, the network is not completely disbanded. I mean, it hasn't disappeared, but it now takes more effort to go from one node to the other, because you have to go through a longer path. And then you have the modularity, which tells you if you can actually separate the network into groups, and I didn't find any difference in any of the conditions. So essentially the modular structure of the network seems to be the same, but there are no additional nodes whenever you want to go from one node to the another. But there is one final twist to this story, and then I'm finished. The final twist is this. The brain, we have talked about the function connectivity of the brain. We have talked about connections between pairs of regions in the functional sense. I mean, if two regions are doing the same all the time. But there are also structural connections in the brain. This is from a very beautiful paper by Hackman and from the Olaf Sporn's group. They applied a method to identify the anatomical connections in the brain between all pairs of regions. And they found essentially the structural connectivity of the brain at a certain scale. This is real connections. This means that there are real bundles of axons going to one connection to the other. And obviously, the functional connectivity of the brain has to somehow resemble this structural connectivity, right? Well, I did this analysis, this is the functional connectivity graph, which, again, these connections here mean that the regions are working together, but you don't know if there is a connection between any pair of regions. You know this from here, but not from here. So obviously the question is, what's the relationship between the functional connections and the structural connections? And there's lots of debate on that now. But for our purposes, what I did was an analysis in which I obtained a measure of how similar the functional connectivity is to the structural connectivity. So I have this network and I have this network here, and I can tell you how similar they are, right? And then, of course, I wanted to know if this changed for psilocybin. So this is the placebo again, and here you have psilocybin after versus psilocybin before, and also psilocybin compared to placebo. So you see that for all this range, essentially, the networks obtained after the psilocybin injection start to resemble more the anatomical connections. So there is a disintegration of the network, but we know also more precisely how the network is being disintegrated. It's being disintegrated because kind of shortcuts between two regions which are not connected anatomically start to being lost. You start to lose them. So the network obviously becomes more difficult to go from one node to the other, right? Because you lose the shortcuts between the regions which are not directly connected in the anatomical sense. So they are not, right, in some sort they're not disintegrated, but they're reduced to their anatomical backbone. So I will end up with this slide in which I will elaborate a little bit of what this means in terms of the order-disorder hypothesis. Last year, well, this is from this year, but last year we started talking with Dante, which is a friend and colleague and has been my advisor, about order and disorder in the brain related to consciousness. But we were not thinking about psilocybin. We were thinking about more drastic changes in consciousness, like sleep and coma and so on. And here you see that you can have integrated and more disintegrated states, and in the middle you have a state in which there's integration, but it's not a complete integration. There are also some sorts of differentiated groups of areas of the brain. And here you have complexity, which is how complex are the activity, how complex is the activity you see in the brain. And essentially these two states, very integrated or very separated, have low complexity. There's in the middle a point in which you find integration and segregation. And we were thinking that essentially during sleep, I essentially work with sleep. I mean, my main area of work is sleep, and I'm starting to work with this data now. Sleep resembles this state in which everything is integrated, nothing very exciting happens. And then I read a paper by Robin and David, in which I struck this figure here, and was very much excited to see that essentially they were thinking the same terms about psilocybin. The hypothesis that also Robin exposed in his last slides is that when psilocybin, you are in a more disordered state in which you can explore more efficiently the different configurations of the brain, but that means that you go to this site, the more segregated. You need to go. If this is true, you need to go to a more segregated state, which is why I somehow have shown with the global connectivity analysis, and also what Robin and the others showed before with these two networks. And then, of course, in the other extreme, you have the less entropic state, which will correspond to sleep or coma, etc. So there is this continuum. This could be true or could not be true. I mean, as all hypotheses in biology, it may be half-truth, but still, it's remarkable that we find so much evidence confirming this, and obviously it's good to have. It's good always to have a hypothesis. Okay. Well, I think that's all. So the final lesson you need to take is that different states of consciousness. Here it says can be. I will replace it by could be understood as displacement from this optimal point. So finally, I want to thank all the researchers involved in the project. I don't know most of them, so I just copied this title of the paper. So these are the guys who made this possible. And actually handed this data to me and Dante to start working a little bit more on it, looking at other aspects of it. Then, of course, I want to thank Dante, Chialvo, and Henrik Jensen, which are two physicists with whom we have very exciting discussions about what all this could mean. And finally, I want to thank you for your attention.
10 minutes for questions. Can I just ask you about, so would you predict, to get back to one of the earlier questions, that there's a hyperstability in conditions like OCD, perhaps? Okay. That's the prediction. Has anyone studied that?
No. That's the short answer. Obviously, people will start...
That would be your prediction.
Yeah. Okay. Predictions about the people, not about the studies, what the studies will say. That's easier to predict.
Okay. Good. Any other comments or questions?
I had a comment and two questions. Thanks for the talk. The comment was that you needn't worry about the hippocampus giving rise to seizures that just cause memory loss, because it does. There is a form of epilepsy where the epileptic activity just sits in the hippocampus and you just get uneasy. And there's quite a lot of work on why it is that when the activity spreads, you begin to lose consciousness, which might be interesting to bring that work into contact with yours. The questions were, I wondered, how do you have shortcuts which go faster than the anatomical routes?
Sorry, shortcuts?
How do you have shortcuts in the brain between nodes which are more efficient, have a shorter path length than the anatomical routes? Because you were implying that you lose those with psilocybin. I just wondered how they work.
Yeah, the answer is that anatomical connections, obviously, when you have two regions which are connected in an anatomical sense, they are most likely going to work together. But it's not trivial that two regions, that you have one region here and lots of different connections to other regions, and you have at the other end another area, right? And obviously the first region is going to resemble more, it's going to do the same as the first region which is connected, and then it's a little bit less, could be a little bit less connected, this first region to another, like a chain in which you start to lose the message. Like this game in which I tell you something and you tell this to somebody else, and in the end it's nothing to do with the original message. And well, this is what doesn't happen when the first region and the last region still work together, right? You are not losing the message. So if you look at functional connectivity, you have the first region and the last region still doing the same. And it could be the case that no, that the message was lost and the first region and the last region are doing something completely different. So that's what can happen.
The other question, comment, was just that your work seems to have quite a lot in common with Tononi's. I wondered if you'd applied his method.
Yeah, of course.
Well, maybe for the rest of us, explain what that means.
Well, there is a very influential theory of how consciousness arises in the brain due to essentially Tononi, Tononi, and he says that the consciousness is always integrated in the sense that you don't have at any time, unless your brain is split, you won't have two consciousness. Consciousness is one. If I clap your hands at the same time and I show you something, these two sensory percepts are integrated in one scene, which is the conscious scene. That's kind of a heuristic view of the oneness of consciousness. But on the other hand, also consciousness is very flexible. Consciousness can support a lot of different states. I mean, think of all the different conscious states that you will have. Right here is a conscious state, but here 1 second later is another different conscious state. So there are lots of different possible states. So he claims that the only way to integrate these two heuristic views, subjective views of brain functions, is to have systems which are separated into modules or more segregated, but they're also integrated. So there's a trade-off between integration and segregation. That's essentially the core of Tononi's hypothesis. And of course, this fits very well with this entropic view of the brain, because if you go to one extreme of this entropic line which Robin introduced in his talk, you will find that everything is super connected. So you have lost segregation and you have only integration, and the other extreme, everything is separated, so you have lost integration and you have only segregation, and in between you have both of them in more or less balanced state. That's why this corresponds to the sweet spot, as he said, of consciousness.
Jan?
Do you have any ideas about the connectivity or your networks in patients with depression or PTSD?
Certainly not. I know that there are papers studying I know that there are papers studying connectivity, and I think that there are some people here, like Robin, for example, who will be more qualified to answer that, because I really don't know how this connectivity changes occur during depression.
I don't know about depression and other disorders in periphery, although psychosis, actually, you may see something similar at Bullman, I think it's done some stuff.
Okay,
but with the infant brain as well, I think you get increased clustering.
Yeah.
I mean, there's more clustering, there's less kind of small world.
Yeah,
In what brain?
The infant brain.
Infant brain, no.
I was not talking anyway about these global measures, but maybe connections inside networks, like the Informal Network, for example. You have depression, you have segregation, you know? Is that right? You lose connectivity between specific networks, but it has not probably been studied, or I don't remember what happens with this global connectivity feature.
We just published a paper on the brain stimulation.
Hang on, give her the microphone and explain who we are as well. Not everyone knows.
Ben Marchant from Amsterdam.
Ah, that was.
He's Jan van Amsterdam from the Netherlands from Amsterdam. We just did a study on the brain stimulation in OCD, and it showed that the improvement of symptoms was actually not so much because the change of the local stimulation at the nucleus accumbens, but by the changed interaction between the prefrontal cortex and the nucleus accumbens. So that might.
Yeah, of course.
Okay, look, we're going to have to stop. Can we just say thanks again to Enzo, please?